In research, one needs vision, hard work, opportunity, and serendipity to play a role. And everybody looks to style their career. You see the people here that have excellent careers around the room and how they're identified nationally or internationally. So one of the issues that I get is why sorghum? So I had an omen, or I saw an omen with sorghum. And it wasn't on a show like Nova on PBS. It was The Walking Dead.
Mapped out some of the agricultural supply places in the area. Even if they've been cleaned up, my bet is that the sorghum would be untouched. That there's a criminally underrated grain that can change the game with our food situation from scary to hunky-dunky. Sustainability, drought tolerance, grain to stover ratio that is the envy of all corns. Think about it.
Nice.
The person on the right works in my lab when he's not on the zombie show. So everybody finds their way one way or another. So think about it. So my presentation today is understanding the carbon allocation networks for sorghum. I serve as a Coker chair. This is a historical landmark in Hartsville, South Carolina. Coker family was one of the families that started the seed industry in the southeastern United States. Their germplasm, cotton and soybean, actually went to Delta Pine and subsequently to Monsanto. So there's connections there. And the family was always interested in linking research, education, and agricultural economic development. So again, in my presentation on this carbon allocation network, I want to break it down into 3 or 4 sections. A background section for advocacy, not only as sorghum as a food crop, but really as a carbon or solar energy accumulator. Understanding genetics and genetic diversity, we have the potential to make it whatever it needs to be made into. I'll highlight 2 case studies from an agronomic genetic perspective, and then lastly I'll present a brief overview of some of the new work that we're doing in DOE project that Mike Gore and Ed Buckler have a Terra project that's a DOE Energy to Fuels project, so we'll talk about that at the end. Okay, seeing myself, this is what I convinced myself I did. I worked in a repository, and I see myself as Noah. Now, I've worked in repositories. Cornell is greatly interested in genetic resources and genetic resources use. And I've worked with numerous of the CGR gene banks. So as a genetic resource scientist in the 21st century, what's different between what I do and what Noah did? Or what capabilities I have that Noah didn't have? So for example, we have automated sequencing, we have computational muscle, we have theories of genetics. His was sort of interesting because he focused on animals and I focused on plants. One thing that always I had to deal with being in the federal government is I also had to know how budgets were allocated and what the real cost of effort of maintaining a particular accession of cabbage or apple would be. I'm not quite sure I had a budget. The other thing that we tried to do was focus on plant genetic resources conservation, but I think until the early 90s, the primary emphasis was on the word plant, not genetic. And I think over the last 20 years, with work that's going on here and in my program, we tried to focus on bringing genetic capabilities to affect conservation and ultimate use. And ultimately, we're not gauged on how effectively we conserve a California condor or a blue whale. Ultimately, the germplasm system in the United States and around the world gets evaluated on use of those genetic resources. So how do you move those genetic resources from a stale old gene bank into actual programs so they advance studies of molecular biology or crop improvement? This highlights genetic diversity in sorghum. The The fundamental question that drives me, and I know it's simple and people probably have plenty of answers to this, but it's what is diversity? If you're working in a repository, you have X number of slots. So how do you fill X number of slots in the repository? So regenerating a cabbage that's a biennial and requires cold temperature before it bolts and is insect pollinated is different than maintaining an apple as a clone in a field or in cryopreservation. So really understanding what the units of diversity are is really important. And the definition changes because it's based on our genetic understanding of an organism. So when I first went to Geneva in 1987, genetic diversity in apple were phenotypes of apple fruit. That was it. There's 35 species of apple. They had 2,800 clones of Malus X domestica and probably 1 or 2 representatives of all the other species. So that's changed. And at the time, they knew 30 lethal mutants in apple. That was their extent of their genetic understanding. But now we have the capabilities and the challenges to think about the insights and tools that we have to build the collections more effectively, particularly for operational and strategic issues. And as our allied sciences in plant pathology or molecular biology or breeding advance, the genetic resources programs need to move aggressively to stay in tune with the programs. Any sort of divide creates inefficiencies and ineffectiveness. So another question is, with genetic resources, whether it's maize or rice, example of sorghum here. These are races, and we'll talk about panicle architecture. How many genes to go and make this transition from the wild type to the cultivated type? And is a lot of genes with small effects, or is it few genes with big effects? And in breeding, obviously, if it would be few genes with big effects, would be better off. But those are the kind of things that we try to resolve and answer, and they're critical in agriculture. So I'd like to highlight some of the importance of sorghum as a background. So it's agronomically and culturally important in semi-arid environments. C4 metabolism, so it's highly productive. It's resilient to potential climate change with water and temperature. Nutrient use efficiency. It produces multiple products. I'll talk more about that. Size and simplicity of the genome. I'll talk more about that. And it's evolutionarily close to things like maize and the other grasses. And there's a building community of scientific support and infrastructure. And lastly, I added that recently. It's zombie-proof too. So there's plenty of advantages for sorghum in the 21st century. This is the big brother and little brother. Similarities and differences between sorghum and maize. So let's talk about productivity. How productive is maize in the United States? What's the average yield? Margaret, I'm not going to ask you that. You're a ringer. It's like 175 bushels per acre. So sorghum had a record this year for yield in the United States. What did sorghum yield? 77 bushels. So how does a C4 plant only yield 77 bushels per acre? There's plenty of complexities. I believe it's more G by E by M. People that grow corn manage corn well and optimize with nitrogen and water. Sorghum's sold on resiliency. So the people that I know that grow sorghum in new areas, they put it on the land that they wouldn't put other crops on. And they don't treat it right. And they'll go out and get 35 bushels per acre. So I think it's really management and the environment, especially if water's not accessible. So although there's a recent divergence between the two taxa, about 12 million years, there's different centers of diversity for maize, New World, and sorghum in Africa. They have wildly different mating systems. Sorghum has a perfect flower. Differences in genome complexity, size, and fluidity. The genome size in maize is about 3 or 4 times that of sorghum. And there are differences in the size of the research community as well. However, I'd advocate, are there any people from Buckler's lab here today? Okay, not yet. I'll get them later. There's really a reason that we need to take a comparative approach linking maize and sorghum and other cereals. There's just real advantages for the simplicity of the sorghum genome to be used to advocate or advance things in maize. So there's a lot of similarities, but there's differences. And there's one big difference. There's a world of difference between maize and sorghum. Sorghum is closely related to Johnson grass. And there's gene flow between the cultivated sorghums and Johnson grass. So that has major implications. So what are the major implications? Cultivated sorghum isn't transgenic. There's no cultivated sorghum that has a transgene in it. There's some in labs where people have worked on it, but now the sorghum community is trying to say, here's sorghum in the 21st century, the climate resilient, non-transgenic, gluten-free cereal. So they're trying to use it as an advantage, but without transgenics and the potential to use transgenics, the world of how you approach understanding and using diversity between maize and sorghum are wildly different. And we'll talk more about that. And particularly, there's more of a focus on, we have to be focused on natural variation. And then it highlights how important genetic resources collections are now, because clearly that's the reservoir for the material to make advances. Okay, so I'll talk about the opportunities and challenges for using natural variation. There were two major advances. Basically, the introductions of sorghum in the 1850s, and it actually came in not as a grain crop, but as a sugar crop. The Mennonites and other religious groups were thinking about substituting sweet sorghums from Africa for sugarcane production, so to reduce the amount of slave labor in the United States. Then in the 1960s, Texas A&M and USDA created a conversion program. And basically, they were taking tropically adapted wild materials from Africa and converting them into photoperiod insensitive, short stature materials for grain production. Unlike humans, sorghum is native to Africa and had a diaspora. So there are 5 major races. And the interesting things is one race went to India and then subsequently came back. You see the movement east and west. That's really easy because you stay in the same photoperiod. This is the interesting move here, because it turns out in this race, Kaffir, and in Guinea bicolor, that's where we find the photoperiod insensitivity genes that are used in converting the tropically adapted to the types that are grown in the United States that mature in 90 to 100 days. So again, by understanding the evolutionary history of these 5 races, it allows us or gives us insights into potentially how to look through collections to find things more effectively. Okay, and this highlights the races. Seed and panicle. Interesting things. This panicle, tight. This wide open. What's the difference and why the difference? Basically, if you're in a place like Mali or some of the spots in sort of west central Africa, it's wet during seed maturity. They don't use fungicides, and grain quality is an issue. So these open panicles, if they're moist, they dry out. The Dura types are types that are used almost exclusively in really dry areas, like Ethiopia, Somalia, those areas, Chad, those type of areas. So panicle architecture is really important from an agricultural perspective, and also over hundreds and thousands of years, there's cultural affinity for certain races. So in sorghum, races are really important and create challenges for ultimately using genetic diversity. Another complexity in looking at diversity, this is from New York. This is a technician who worked with me, Charlotte Acharya. She's now at UC Davis working with Steve Knapp. But besides racial types, there's types based on use. Here's grain sorghum, short-statured, sweet sorghums that accumulate sugar in the stem, like sugarcane, and then these cellulosic biomass types. Total carbon per unit time, per unit area, per unit input. They're photoperiod sensitive, so you plant them in the United States and they grow until they freeze. So what we're looking at here is total carbon that can be converted, and I'll come back to that in a bit. So here's an example of size of some of the ex situ sorghum collections. The big collection is at Kresovich in India with over 40,000 entries. The National Plant Germplasm System has about 42 to 44,000 now. The Indian National Program, the Chinese program, and there are other intermediate-sized programs in other countries in Africa and South Asia. Totally, there's probably 170 to 180,000 accessions. But as a geneticist, what's the level of redundancy in there? For a long time, India and the United States were trying to make sure there was an appropriate backup. So there's probably a lot of redundancy between the two major collections. And the significance about the collections is they're big, they're uncharacterized, and it's unclear how diverse they are. Another thing is the politics of global germplasm. It's difficult at times, or impossible at times, to move germplasm. We all know stories about rice, soybean, coffee. So moving materials is a real challenge these days. So having an effective way to really go through our National Plant Germplasm collection of 40,000 is really important. And this highlights this. And the key factor, the key thing I want you to look at is right here. This 25% of 40,000 is photoperiod insensitive, which means it'll grow and produce a panicle in a certain number of days. This is the 1,000 that were converted from 1960 to about 2010. 3/4 of the collection are these big giant types that don't have the agronomic phenotypes that people are interested in. So what we've been trying to do is spend our time up here. It's readily accessible. Griffin, Georgia is about 2 hours from where I work. So, and again, to give you the complexity, to highlight this, this is an example of the conversion program. This line is this line converted. 4 genes associated with height, 4 genes associated with photoperiod insensitivity. Everything else is the same. Okay, in parallel, changes in technology affect biology and agriculture. So I was at Geneva, like I think Jeff Doyle will remember, this is like prehistoric time. I was at Geneva for 4 years, and then I won a federal fellowship. So in 1991, 1992, I had the opportunity to go to WashU and worked at WashU. I spent time with Barbara Shaw and Alan Templeton in population genetics, but I spent the other part of my time in the genetics group. And at that time, it was like Maynard Olson and Dan Hartle and Rick Wilson, and it was ultimately the place that became the Washington Genome Center, Washington University Genome Center. And at that time, it was clear to me that we needed to better genetically characterize a collection. Now you know with DivSeq and some of the other groups, there's questions about sequencing collections and how one might do that, what the biological questions are, and how you target what you sequence. But ultimately, our capabilities in genetic characterization is going to allow for better management and use. And there's been great progress. Here's an example. This is an outcome. I wanted to highlight some of the advances in genomics developed in sorghum. So in 2005, the NSF sponsored a genomics workshop in St. Louis. There were about 40 or 50 people. And we put together a joint Genome Institute community sequencing project at that time, and it led to this publication that's still widely cited, Sorghum Bicolor Genome and Diversification in Grasses. Andy Patterson was the lead on the proposal and also on the program itself. That set the tone. After rice, sorghum was one of the important cereal crops that we had genomic information. But at that time, it's one reference line. The current well, I'll talk about that in a second. There's Let's go to the next one. This is from a really good group in Queensland. Emma Mace, David Jordan, and I guess Ian Godwin are sort of the three pushers. They work with people in China to do whole genome sequencing, and they have about 50 lines that were resequenced that represented genetic diversity. But what I wanted to highlight now is probably between a number of programs, there are 500 lines that are resequenced now in sorghum between groups nationally and internationally, and I'll show that in a second. The last thing I wanted to highlight, and this came out of work at Cornell, was a bread project that was funded around 2009. And Jeff Morris was a postdoc with me. He's now faculty line at Kansas State, but we worked with Ed and Ed's group and Sharon Mitchell and a number of other people to develop GBS data in sorghum. And just to give you an idea of what that resource is like now, there's probably somewhere between 5,000 and 7,000 lines that we have GBS data on. That's been for multiple years now, and we use it regularly in our lab. And each accession was genotyped for about 400,000 markers. Depending on the biological question you're asking and your level of confidence or discomfort, you may use less of those markers. But the size of the sorghum genome is around 700 megabases. So with 400,000 markers, on the average, we have a marker of about every 2 to 3 kb, which is very good. Also, generally or average speaking, linkage disequilibrium is about 15 to 20 kb in sorghum. So we've got plenty of resources, maybe not everything that we need, but there's plenty of resources there to take advantage of. And again, we're starting to work with groups internationally, CRAD in France, the ICRASAD group, the Queensland group, and basically all these programs have somebody in sorghum that's either doing GBS or resequencing right now. So there's a significant number of groups involved. No, it's not maize. No, it's not soybean. No, it's not rice. But there's a lot of resources there. So I'll stop here and start to talk a little bit more about how the resources can be used. Okay, a key question to me over the years, I started out working in sugarcane and sorghum. A key question to me is productivity affected by both source and sink strength? Which plays a bigger role and under what conditions and what mechanisms? So I'm going to highlight a little bit of a tale of two crops, sugarcane and sweet sorghum. What my ultimate goal is, like the next time I give a seminar, I want to come and I want to be introduced as the master of carbon in plants. That's my goal now. That's what I want. I'm not there. Okay, a little bit about sugarcane. Sugarcane diverged from sorghum about 8 or 9 million years ago, and it's impossible to work with. When I did sorghum or sugarcane breeding early in my career, our strategies for crop improvement were cross the best with the best and hope for the best. So and also, it has somewhere between 100 and 120 chromosomes. It's an octoploid. You vegetatively propagate it. It has limited range, requires water. It's a very complicated crop. The thing to contrast with sorghum is carbon accumulation pattern. So I'll start with explaining sugarcane. Sugarcane in commercial production, they don't want flowering. They assume that the flowering would be transition of carbon that's stored in the stem to the flower. So there's an industry of Monsanto and Syngenta and other companies that produce compounds that inhibit flowering in sugarcane. And it's used in a lot of countries. And they feel that it increases the value of the sugarcane because there's more sugar per unit stalk. So in sugarcane, we don't want flowering. Also, sugar accumulates from the lower inner nodes up. It loads at the bottom and then it loads upward. Okay? Now sorghum, close relative. Let me go back. So a close relative, and questions that affect carbon partitioning are the fundamental distinction between sweet and biomass types. First, we have photoperiod sensitive things that don't flower and things that flower. The genetic mechanisms remain largely uncharacterized. And whatever we do in carbon affects all the different types of sorghum. If you want to allocate it to more leaf or a forage type, you can do that. If you want to produce grain, you do that. If you want to produce a crystalline sugar, you could do that, or total carbon per unit area. But the interesting thing about sorghum is, and this gets back to the source-sink relationship, sorghum does not accumulate sugar in the stem until it goes reproductive. So it has small amounts in the stem, but it doesn't accumulate. So it's just the opposite of sugarcane. The other thing is, an accumulation pattern is just the opposite of sugarcane. It loads from the upper leaves downward. So I'm not an evolutionary biologist. I'm going to look at you, Jeff. They're so close, and they have wildly different patterns, which is curious because you don't get any sugar accumulation, and that leads us to believe the power of sink strength in sorghum because you're filling grain the same time you're loading sucrose or simple sugars in the stem. Okay? So this is from our field in South Carolina. This is like really sandy soil. This is, the ocean used to be in the middle of South Carolina. But this is where we work because with our DOE Bioenergy project, we're trying to look at marginal lands. And these are the criteria for a bioenergy idiotype that we work toward. Desired maximum efficient capture of carbon via photosynthesis per some unit. Could be time, land, water, production input. And look at the yield that we get. Our goal is 15 to 20 dry tons per acre. So Don Vians isn't here, so I can't agitate him about switchgrass. But you could see this grew in about 4 or 5 months. And the problem there is lodging, obviously, but that shows you how productive the material could be. So in the summer, July and August, you could see a growth of the plant like a foot in less than a week. So it grows when it gets warm and hot, or gets warm and there's moisture there. We're trying to make sure that most of the carbon goes into the stalk, and we hope that the leaves will be recycled to improve the soil quality. We work with J-Bay and out in California. The things that we take back from them is they want low lignin, low minerals if they combust it, because slag in an oven or when they're generating electricity would be a negative thing. And we don't want to haul around water. So we breed for dry stems and dry down as fast as we can. And the other thing we look towards is, it looks like C6 carbons are way more efficient than C5. So that's what we select for. So we do a lot of compositional analysis in our work. And then just resistance to critical biotic and abiotic factors. Okay, so this starts to highlight Zach Brenton's work, who's the bioenergy person. And I want to highlight, again, this connection between genetic and genomic resources. It's one thing to go up to you and say, here's seed from the seed bank. I would rather have someone to give me that, and on the other hand, give me like some information that I could use to really exploit that seed that's going to come. And I think there's going to be more and more need, and I think one of the take-home messages for the presentation is like, for people that do genetic resources conservation or plant genetics or plant breeding, as molecular biology advances, your contributions become more and more valuable. People want to work with neurocygenic lines, or they want diversity, or they want contrasting phenotypes to do the mapping work. Somebody has to take raw genetic resources in a repository and mold them to fit particular biological questions that are going to be asked. So again, we're trying to fill that niche with some of the work that we're doing. So in 2008, we developed the first sorghum association panel here. Alexandra Costa was the lead on that. And subsequently, we've created a bioenergy panel, and I'll talk to you about that. The question I would have is like, you already have an association panel in sorghum. Why do you need this bioenergy panel? And I hope to talk about that now. So first of all, whether it's the sorghum association panel or the bioenergy panel, we capture a lot of diversity. Race is important, but there's plenty of admixtures, so you can see how the races group here on this figure. Okay, so races are important, but admixture occurs. The other thing is we have this mega data set. Almost everything that we work with in the program is GBS. So frequently, we wind up helping people by providing germplasm for people that are setting up genomic or molecular biology or genetic or breeding studies. So we work from a data set of 5,000 to 7,000 and then target it appropriately based on the biological question that the investigator is asking. So do they need diversity? Do they need particular phenotypes? And that's a thing that we spend a lot of effort on. What did we learn from Mays and Buckler? Too bad he's not here, this would be good to agitate him. What was bad about the challenges with GWAS? Confounding variables. Height, flowering time, low phenotypic frequencies, and population structure, right? This highlights in sorghum maturity 6 gene that you see doesn't flower. So again, I see from Mays what the challenges were, and what we're trying to do is work on particularly affecting the field experimental design and phenotyping, whether it's height, maturity, or whatever, and then the panel size that we're investigating. So what we found in the bioenergy panel, or one of our driving factors, I was with Jiming Yu and we were talking about the complexities one time. So how we get around the maturity pattern is we use photoperiod sensitive things. You plant them, they all grow the same length. They're all the same developmental stage. There's no maturity issues. So we've overcome that. We were able to establish genomic diversity relative to the original sorghum association panel. And since we had phenotypic data, we put in things that were biomass related where we would first look at genetic diversity and subsequently overlay with phenotypic data. So in an instance where you could pick from 3 or 5 lines, which were the most intriguing phenotypically to provide contrast. So that's how we worked through that material. Okay, and this highlights a process. First, the collection has 40,000. We acquired over 4,000. From the 4,000, we got the Bioenergy Association panel of about 400. And from that 400, we picked parents to set up a nested association mapping population. But in not only using diversity, we tried to look at phenotypic differences and real changes. So look at the range of characteristics. In bioenergy, yield is important, but composition is also important because it goes back to C6 to C5 carbons and lignin. So here's our dry matter yield in the panel, 3 to 20 tons. Percent fermentable sugars, 3 to 21%. And you can see cellulose, C6, C5, and lignin. So we've got significant ranges in those materials. So again, we're trying to use our genetic insights, but overlay phenotypic information on that. And again, I received a grant through the DOE Feedstock Genomics project or program to develop a NAM population. So if any of you were associated with that getting funded, I would like to talk to you subsequently because I'd like to kill you. I propose to do NAM parents that are photoperiod sensitive. So I actually thought we could get, by doing tricks to induce photoperiod, to induce flowering, we would put trash cans on the plants when they were like 2 feet tall in South Carolina for 2 weeks, and then take the trash cans off to see if we induced photoperiod, or to induce flowering. It worked 1 year. So the other years, basically, with the NAM populations we were working in, we were only able to get 1 generation a year. So we have, next year, there'll be 14 or 15 populations that are completed, and they'll be to F6. So it isn't a perfect world, but it's material that fits the DOE program and should be useful. And we'll continue to work with that. But it was just a real challenge, and it's taken a lot of effort. Okay, so how do we use it? This is from Zach's work. Zach's particularly interested in gene pool studies and targeting traits, and one that's important is non-structural carbohydrates. So that's sugars and water-soluble carbohydrates. So this is a preliminary analysis over 2 years. So we found some interesting regions of the genome that correlate with 13 loci, 20 significant SNPs, and 65 genes in that 20 kb window. The 20 kb window is the average value, and that's why we use that. We're working on trying to develop more specific LD in particular regions. Okay, so we're off to the races. So the 65 genes that were identified, 20 are graph specific, 25 have no function, and basically there's limited annotation on any of those things, so how are we advancing? This might be expected because the Poaceae, Andropogone, or Saccharini, this group with sorghum and sugarcane, has unique cell wall types. It exhibits C4 photosynthesis, and sugarcane and sorghum have a unique phenotype. It's accumulation of sugar in the stalk. It's not grain. There's plenty of work that's been done on grain. So biomass-related traits in general are very poorly understood because really a lack of relevance to other plant models. So we know much more about grains, but there's limited information in sugar accumulation. Okay, so we continue to work, and we found some interesting candidates that we're exploring now, and I'll highlight this in the next couple slides. Here's a vacuolar transporter that looks very interesting. There's also genes associated with cellulose production, sugar transporters. So a number of the things here are sort of round up the usual candidates. What we found with this vacuolar transporter, the NFC stands for non-fibrous carbohydrates, so it's the non-structural carbohydrate. And these are the ranges of difference between these 3 entries and the different haplotypes there. What we found is in this region had a positive effect on accumulation of non-structural carbohydrates and a negative effect on structural carbohydrates. So obviously it's important in partitioning. Rick Boyles works on grain. He also detected this same peak for an effect on grain on acid detergent fiber and neutral detergent fiber in the sorghum association panel. So we've got data from both the Bioenergy panel and sorghum association panel that suggests this is interesting. And again, this is a close-up of that. The highest peak in the GWAS for non-structural carbohydrates is associated with an amino acid change in this vacuolar transporter, and each red line is an amino acid change in the transcript. So where do we go? We go where you all are going and have been going for years. QTL analysis and segregating populations, candidate gene sequencing, and we're also in commercial industries, almost none of the crop improvement programs look at inbreds. They look at performance in hybrids, so we have to do that as well. So again, to move these alleles into useful hybrids, we have to look at their effect in hybrids and not just inbreds. Okay, I'll switch gears quickly to Rick Boyle's work on grain sorghum to contrast what he's been doing. So he's focusing on total carbon, but in the grain. I get in trouble when I show this slide, especially with people in the United Sorghum Checkoff Program. They're in Lubbock, Texas. And almost on all the DOE or USDA proposals I write now, I write something about the new sorghum bioenergy belt. To me, the new sorghum bioenergy belt with climate change is going to be Mississippi Valley, Gulf Coast, Atlantic Coast, where there is water and maize doesn't grow well because it doesn't do well on those coastal sands. So Rick's goals are really to look at grain yield and grain quality. Two approaches, sorghum association population or panel, and then two recombinant inbred populations and a recurrent parent that has a waxy phenotype. So we have data completed for both of these two approaches to understanding what's going on there. His primary emphasis are energy yield and grain quality traits, and that just highlights an example of differences in the panicles. Going back to all that diversity in the Bioenergy Association panel, here's examples of how diverse our Sorghum Association panel is. So here are traits listed. In particular, the top one is grain number per primary panicle. So the range goes from this year, 2013, from 63 to 4,000. 1,000 grain weight, how big the seed is, from 5.4 to 40. Yield per primary panicle, 3 to 125. And then there's compositional characters that we collected as well. And like the next year, you see similar things. So you see great variation and great heritability there. The thing that's intriguing about this is we intentionally overlaid phenotypes on the Bioenergy Association panel. With the Sorghum Association panel, we didn't really. This is just what we got. So we lucked out. So here's our schematic to go from GWAS to candidate gene selection. You have here grain number per panicle, yield per primary panicle, and 1,000 grain weight, and how we went through a process to eliminate things. So eliminating SNPs that fit into the range for LD. And then we also looked at expression data, both in sorghum and maize where we could find it. And this is an example of grain yield per primary panicle. And you could see, we look at multiple models. One of the things that I feel we lack in our program is we always are trying to get more and more computationally literate and look at approaches to analyzing the data. So we're using packages that others have developed. But again, we're trying to look at it as many ways as we can. So this highlights potential areas of interest in the genome for grain yield per primary panicle. This highlights a couple things. Grain number per panicle is associated with something on chromosome 10. Okay? And this is 1,000 grain weight, chromosome 1. So what we're finding is they're independent. So a number of people believe that you have X amount of carbon and it's how you partition it. You either get more seeds that are smaller or fewer seeds that are larger. But there is play in there, and it's likely that you can get more seeds and not so small seeds. So it highlights their different mechanisms. And then we use expression data to see where these genes are active. And you can see it's like in the inflorescence, and in here it's the embryo and endosperm, or in the endosperm here. So again, trying to overlay expression data with the sequence data. And these are, Rajan Sikone was at Wisconsin. He now works at Clemson. He's next door to me, and he's a maize person. So we're trying to get together and look at data sets together. Here, looking at expression patterns, the main one to look at is this ethylene receptor and seeing the difference between large seed in the recombinant inbred population and small seed. So in a sense, maybe this region has a gene that functions as a negative regulator. So we moved on further, and we're looking at a gene called RCD1, radical-induced cell death, and its effect on 1,000 grain weight. So We conducted the analysis in the association panel. And then we've been subsequently doing candidate gene sequencing and looking at expression profiles. So what we're finding is that there are things that match up and make this region or this potential candidate gene look interesting. Here's some of the sequence data. We sequenced 24 lines. The bad news is we get a lot of haplotypes, but the interesting thing is these haplotypes are associated with large seed. These haplotypes are associated with small seeds, but we had 2 recombinants in the middle that actually had the highest grain yield. And we've been looking at it, and we've seen that there are 2 missense mutations and 2 splice variants identified. So again, we're pursuing that. It would have been nice to have less haplotypes and clear discrimination, but that's where we are at this point. We're uncertain of the causal variant, but we're continuing to investigate that. The other thing that's interesting about RCD1 is when you look at it in other organisms, whether it's rice or barley or Arabidopsis, it plays an interesting role in seed size. And here's our model in a sense. RCD is a possible upregulator. It's highest within the developing endosperm, or the expression is highest. In other plant species, it interacts with a wide range of transcriptional factors. In these mutants, they have shrunken seed in Arabidopsis within the stalks, and the maize RCD1 expression is elevated within the endosperm of large kernel types. So in a sense, there are 2 ways of improving grain size or grain weight. You increase the fill rate per unit time, or you expand the grain fill duration. So what we think is RCD1 may regulate expression of 1 or more of these enzymes that are in this starch synthesis pathway. And in summary from the 2 projects that I just highlighted, one of the things is utilizing multiple and/or larger panels for GWAS. So whether it's a regional panel or built on some other things, really looking at ways to minimize spurious associations. Our panels now that we're doing this summer are 4,000, not 400. And we have the phenotype, the limitation on that is actually phenotyping capabilities. So we're doing over 4,000 lines, and they're all GBS at this point. And in addition, we're looking at nested association mapping populations coming online, regional diversity population panels. So we're looking at multiple ways to look at map small effect QTLs. We could get the big ones. We're looking for smaller effects now. And hopefully some of these validations of candidates will yield some insights on whether we're going down a dark alley or not. And corroborating evidence from multiple sources is required. So one last thing, I want to return to the value of integrating genetic and genomic resources. Again, in summary, as great molecular biology in the future is going to occur, they need things from breeding and genetics programs. Significant genetic and genomic resources, diversity and contrasting phenotypes, near isogenic populations. There's all kinds of things to invest time in to reduce noise in some of the things we do as we're working on gene discovery. So I really see a bright future for people in genetic resources and plant breeding. And this highlights some of our ongoing work. So we're going to have a reference sequence with Rio. That's the archetype sweet sorghum. So we'll have BTX623 and Rio. We just submitted a letter of intent to the Joint Genome Initiative community sequencing projects to produce reference sequences for 50 more sorghum lines. And because sorghum is a flagship for the DOE, we think we have a good chance to get that. Through the Terra projects, we have one other transcriptome study going on. We've created near isogenic lines of Rio, and then a Rio that has a dry stalk and doesn't accumulate sugar. So we're doing expression studies there. We've been working on multiple magic populations. So we do grain, male magics, female magics, which is important in hybrids, and then we also do sweetened biomass. As part of our Terra project, we're going to have resequencings of 600 to 800 more sorghum accessions. In fact, 400 are done now. Todd Mockler and Jeremy Schmutz have been working with us on that. And then what we're trying to do is put out hybrids for people to evaluate themselves. So I've got 2 more slides I want to talk about. This is our Terra project. We're working with Carnegie Mellon and a company called Near Earth Autonomy and the Donald Danforth Center. We got $6 million for 3 years to do a project that like Bicolor, Gore, and others are doing. And it's the new opportunity, and it's also the flavor of the month. I'm really skeptical, but the DOE is really interested in bells and whistles associated with high-throughput phenotyping and computational power. The emphasis of our project is on genetics and breeding, but it also has a big element of robotics and sensing systems. So there's actually some Cornell people that are working with Carnegie Mellon, and they worked on the Mars rovers, undergraduates here, and they're designing robots to go in the field and actually to go up to plants and sample plants by using a penetrometer-like thing. Lord knows if it's going to work, but the question becomes, is more data good or bad? Is it like terabytes or terabytes? And I really think we need to think about that. So what I ask our engineers is a real simple question. It's like, what's the biological goal of what we're trying to do when we do the phenotyping? And I really see 2 approaches to advancing genetic gain. There's 2 ways to do this. One is to use biological hypotheses and then use high-throughput phenotyping to follow those traits. More rec leaves, higher density planting, photosynthetic rates, things like that. The other approach is like the engineering version of genomics. It's like measure everything you can for as long as you can, and you throw it up against the wall and see if anything sticks. So in that case, it's really the engineering capabilities, no biology. But we know people in genomics have made projects by big data sets too. But again, I see the value of sort of the high throughput phenotyping for things when you have robots and sensing in the field where there's an invasion of insects or something like that, or drought stress. But it's a more tenuous relationship between affecting yield in a breeding program or affecting genetic gain. So what I'll say to our engineers, I give this to them every time I see them. I say, what are you measuring, and how's it going to affect this equation? Are you going to give me, obviously shorter cycles is going to be useful, so anything we can detect sooner and faster and get more generations per year is important. But how does the engineering approach affect things like that? And that's how we're really trying to work the program. It's actually been really hard at this point because it's more, we're still wrestling with, is it like a engineering thing where they're going to measure everything for 6 months and we try to make sense of that data. We estimate it right now, though, by the end of the summer, we should have over 50 terabytes of data. So good luck. Okay, so I'll finish with that and reinforce one thing. Don't forget about sorghum. If it's zombie-proof and it's criminally underrated, there's potential there. And then also, it's great to be back. I want to thank all those that have had the opportunity to get me back here. Over the years, I've had great collaborations, both with across the United States and across the world. And I've had great people. A lot of the faculty members know the people that worked in our labs on and off. So the team at Cornell was just top notch. I mean, can't get a better group. And lastly, funding sources. Public sector, private sector. We work with companies. We work with, for you get people that are graduate students or postdocs, I look at grants and contracts like you're building a 401k. It's like sometimes the stock market's going to go down. What are you going to have then? So we try to find our program where we find multiple sources of funding to answer things. And okay, one last thing. It was a joke to begin with, what I've learned. One particular thought for the limited, I don't know how many are graduate students here or postdocs. Cherish the time that you have, both opportunities and challenges that you have at a place like Cornell. Take it from me, it's a really special place. When you go to other places and see things, your eyes will light up. So you'll look back and count these times as really special times. So knowing a priori that they're special, take advantage of them now. Okay, I quit.